In this video, we'll be talking about the basic aspects of a DNA, that is the general structure of a DNA, and an outline to the discovery of DNA. We'll be discussing about both these. First, the term nucleic acids, because both DNA and RNA both constitute a nucleic acid. And what does a nucleic acid consist of? A nucleic acid consists of a nitrogenous base, a pentose sugar, and followed by that a phosphate linkage. This is what is the basic component of a nucleic acid. It contains nucleotides. So we have seen about the individual aspects of each and every individual unit in the previous video. Now first, this nucleic acid was discovered by Friedrich Mischer. So he was the scientist who identified the nucleic acids. And where did he identify? He identified in the pus cells. So what are the pus cells? The pus cells are basically the dead cells, but they come out from a living tissue. For example, when our skin is wounded, we leave it open for some time. And if we get a secondary wound on the area where we are hurt, then it may open up. And through the platelet tear, all these cells may urge to push out. That is called as a pus cell. And he identified these nucleic acid in the pus cells. And he also told that the DNA were basically acidic. So he classified the DNA molecules, that is the nucleic acids, as acidic molecules. Then the term DNA, because at the time when Friedrich Mischer was working, there was no term called DNA that was actually given. He had just been calling it as nucleic acid. Later, it was Zacchaeus who gave the term DNA to the nucleic acid. And later on, further research, the RNA came and many other such. They were making some internal research of the structure of the DNA, RNA, many things. We'll be seeing about it. But at first, the term DNA was given by Zacchaeus. But the structure of the DNA is still not known. So the structure of the DNA was proposed by Watson and Crick, and for which they were awarded the Nobel Prize as well. So basically, the nucleic acids were first discovered by Friedrich Mischer in the pus cells, and he told the nucleic acids are basically acidic. Followed by that, the term DNA was given by Zacchaeus, and the structure of this DNA was proposed by Watson and Crick. Now going into the structure of DNA, what is the structure of DNA? What does it contain? I already told you, DNA is basically a nucleic acid. A nucleic acid means it's a polymer of nucleotides. What is a polymer? A series of repeating units. So you have one nucleotide, followed by that another nucleotide, followed by that another nucleotide. A series of nucleotides together, a polymer of nucleotides is what is called as basically a nucleic acid. So DNA is a polymer of nucleotides. What does the DNA contain? All nucleic acids contain the same thing. It contains a nucleotide. So a nucleotide in turn consists of a pentose sugar, a nitrogenous base, and a phosphate linkage. These are the three components of a nucleotide. But when I talk about a pentose sugar, it is going to be of two types, a ribose sugar and a deoxyribose sugar. Since this is DNA, here we have deoxyribose. In the same way, nitrogenous base. There are purine bases, there are pyrimidine bases. But when I'm talking about DNA, it is going to be adenine, thymine, cytosine, and guanine. And this is going to be substituted by uracil when you come to the RNA, which we'll be seeing in transcription. But basically, the nitrogenous base is of four types here: adenine, thymine, cytosine, and guanine. And apart from that, you have a phosphate linkage as well. So this is the basic structure in which the DNA is going to be present. Now, when I'm going to draw the structure, let us see now. Now, this is going to be the phosphate linkage. And this phosphate linkage is being attached to the fifth carbon of the sugar molecule. And this sugar is nothing but deoxyribose. And this deoxyribose can be identified with H and OH here due to its stability. And to this side, the sugar first carbon is being attached to the nitrogenous base. If it is going to be a purine, it is going to be attached to the 9th carbon. If it is going to be a pyrimidine, it is going to be attached to the first nitrogen. So this nitrogenous base is going to be decided either as a purine or pyrimidine based upon which nitrogen is going to get attached to the first carbon. Now, how does the chain of nucleotides get formed? Now, it is very important to see here that there is a OH functional group here. At the third carbon, there is a OH functional group. To this OH functional group, the new nucleotide will get added. The new nucleotide means another set of nucleotides. This is one nucleotide. Let us name it as 1. There will be a second nucleotide which will be connected to OH, and that will have a free OH. To that 3 will be connected, and that 3 will have a OH. To that 4 will be connected. This series of nucleotides together will form a DNA polymer, or that is together called as a polynucleotide. So a DNA is a polymer of nucleotides. So it will contain 3 basic structures: the pentose sugar, nitrogen base, and the phosphate. And the pentose sugar is the deoxyribose. Nitrogenous base is adenine, thymine, cytosine, and guanine. And the phosphate will be connected to the 5th carbon, as we already saw. And all new nucleotides will be attached only to the free functional group at the 3rd carbon, that is the OH plus.